today about this disease, multiple sulfatase deficiency, and our efforts in trying to move the science and the medicine forward for this. But before I start, I really want to say that the only reason why this disease is being represented in this group is really the work of the advocacy partners that we partner with very closely, both the US-based advocate group, which Amber Olson is here representing, and also, yes, I agree, Bob. And our European partners and our European colleagues, really spearheaded by Ahrens-Nicklas in Ireland. And they have really taken it upon themselves to move the science and medicine forward and helped us partner actually with the GLIA initiative here. And it's really the infrastructure of the GLIA initiative that allowed all of this to happen that we're going to be speaking about today. So thank you to lots of people before we even start. So our topics for today, I'll do a little bit of an intro. I'm a physician scientist here at CHOP and a metabolic physician that cares for kids with lysosomal storage diseases. And then I'll pass it off to my colleague, Dr. Lars Schlotawa, who's visiting us here from Germany to talk about some of the key successes in developing therapies. And then Dr. Laura Adang, who will talk about some of the challenges. And they're really intertwined as we were all making our presentations, we realized you can't talk about one without the other because it's really driving our work forward. We had a great discussion in our work group. Here's a roster of our members. And I think what's really unique and interesting about this disease is that the buy-in from the scientific and clinical community is huge. And so the foundation leads have really found people around the world who are passionate about trying to improve the quality of life for kids with this disease. And you can see here, we go all the way from basic biochemists who have thought about the structure of FGE for years in a test tube or in a plate, all the way through to clinical research experts. And so it's really the buy-in of a multidisciplinary community that have made this all possible. Okay, so a little bit about multiple sulfatase deficiency. It's a progressive multisystemic disorder that arises from mutations in the enzyme that's responsible for the post-translational modification and activation for all cellular sulfatases. So sulfatase enzymes are enzymes that are important to catalyze a variety of different reactions within the cell, but they all start from the same place in that you have your sulfatase translated in the ER and then post-translationally modified. So basically there's an active site cysteine in all sulfatases that needs to be converted to a formylglycine residue in order to be active. And this is done by an enzyme called FGE, or the formylglycine generating enzyme. Then, once those sulfatases are activated, they can leave the ER and go to a variety of different cellular compartments and serve their function. You can see there are lots of different sulfatases, 17 within the cell, some of which are familiar to this audience. For example, in terms of lysosomal sulfatases, arysulfatase A, the enzyme responsible for MLD, is one of our sulfatases. In addition, there are multiple different subtypes of MPS disorders that are sort of encompassed in this disease. So iduronate 2 sulfatase is the enzyme responsible for MPS 2. There's forms of Sanfilippo or MPS 3 subtypes, and so forth and so on. So in reality, what happens in a patient is that when you lose the activity of this FGE because of mutations in the gene that encodes it, you end up not having all of these sulfatases activated throughout the entire cell. So in some ways, you can think of the pathophysiology of this disease as the combinatorial effect of multiple different single sulfatase deficiencies, hence the name. So patients have features of MLD, of MPS 2, of MPS 3A and D, and so forth and so on. In addition, there's a number of sulfatases that exist in the cell that don't have a corresponding monogenic disorder yet, but we assume that the loss of these activities are also contributing to the really complicated phenotype that our patients have. So, like I said, this is an autosomal recessive disorder. It's due to biallelic mutations in the gene SUMF1 that encodes that FGE protein, so that activator of sulfatases. To diagnosis, we always rely on biochemical confirmation of the disease. So you have to have low activity levels of at least 2 sulfatase enzymes. In addition, some of the substrates that of those sulfatases can build up. And so you can have biochemical confirmation of, for example, novel variants in the gene. There's a wide spectrum of presentation in this disease, and you'll hear a little bit more about that, where children can present in the neonatal or infantile period, or historically it's been thought all the way through the juvenile period. But if you look carefully at those kids' medical records and you look back, they often had symptoms that were sort of underdiagnosed earlier on. And then, like I said, it's a multi-systemic disorder, very similar to what Dr. Adang was speaking about this morning with AGS. It's not just the brain that's affected. In reality, these kids have multiple different MPS subtypes, so they have all the symptoms of those disorders. So just to sort of show that in a nutshell, you have loss of the SUMF1 gene or expression, or often it's actually missense variants in the SUMF1 gene, that leads to decreased activation of all cellular sulfatases, and therefore you get buildup of substrate. And for example, you can have a variety of different glycosaminoglycan substrates that build up. In addition, you can have sulfatides that build up from as you do in MLD. And it leads to multisystemic disease. So of course, global developmental delay and regression are a key part of this phenotype. But in reality, you have eye problems, bone problems, heart, lung, skin, all the things that you would think about for other disorders like MPS layered on top of this. And it really contributes to the morbidity and mortality of patients. Okay, so we'll focus a little bit on the challenges and successes today, both of research and clinical care. Biggest challenge for MSD, but I also say one of its biggest strengths is that it's ultra rare. So the estimated incidence of this disease is about 1 in 500,000. So it's definitely probably one of the rarer ones that we have here today. But that has sort of really galvanized the community to really have a strong advocacy bent and a really strong advocacy aims so that they can find patients throughout the world that are diagnosed with this and offer support. And so a key challenge is optimizing these resources, these advocacy resources, and make sure that they're equally available to patients throughout the world. Another challenge is disease heterogeneity. And so we know that amongst this ultra-rare disease, different patients can look very different throughout the course of the disease process. And so capturing this full heterogeneity in this ultra-rare disease is challenging, but doable, I think. And then biomarker development. I will say this is one of the sort of successes and one of the ways we can borrow from the rest of both the leukodystrophy community and the lysosomal storage disease community. Because we know what sulfatases are abnormal, and because there's biomarkers for a lot of the disorders associated with those sulfatases, we really have the ability of borrowing biomarkers from related disorders, which has been a real benefit to us. In terms of clinical challenges, so clinical care is obviously impacted by the ultra-rare nature of this disorder, right? So most physicians throughout the world that diagnose MSD have never seen a patient with MSD before. And so accessing good clinical care guidelines is really important. Making sure those are up to date and available for families is really, really important. Newborn screening for this disorder, again, we might be able to piggyback on the efforts of some of the related disorders, whether that be metachromatic leukodystrophy, MPS II. Some of these patients might be identified through newborn screening for other disorders. A relative strength, like I mentioned, is our clinical care guidelines that we really developed at request of the advocacy groups in order to help uniform the care that patients are getting around the world. And unfortunately, there are no currently approved therapies, but people are hard at work on this, and you'll hear more about that today. So here's our heat map. And first of all, I just want to take a minute and say how fortunate we are to even have a heat map. As we were preparing this talk, Amber, who's sitting in the audience somewhere, texted me, she's like, we have a heat map this year, right? So the fact that we're this ultra-rare disease has this much exciting work behind it that we could actually start quantifying where we are is huge. As you can see, some of our strengths include the preclinical scientific development, and we'll talk a little bit about that today. There are still definitely challenges out there, including therapeutic development. While it's ongoing, we're definitely not in clinical trials yet. And so I think there's a lot of room for growth. And hopefully this will be even more blue and green the next time we meet again. So with that, I will pass it off to Dr. Lars Schlotawa.
Yeah, thank you very much, Becca, for the kind introduction. Thank you for listening to this. So my name is Lars Schlotawa. I'm working in Goettingen in Germany, and I've been working on multiple sulfatase deficiency for quite a while, and would like to use this talk to talk about developing therapies for multiple sulfatase deficiency, and also kind of wrap up what's been done in the past and what's on the horizon as future therapies. So the first description of MSD was in 1965, so we're going back right to the beginning, when Austin et al. Published MSD as a form of metachromatic form of diffuse cerebral sclerosis, so an underform of MLD, And it took nearly, well, it's difficult to calculate, a long time that to discover the sum of one gene, which is also an interesting story if you want to read the literature behind it. But then in quick succession, we had people were able to resolve the crystal structure of the formylglycine-generating enzyme, as well as the development of the sum of one knockout mouse in 2007. And I would say by the time, all the tools that were necessary for developing therapies were there on the table. So we had the gene, we had at least a basic on even for some aspects an in-depth understanding of the pathophysiology of MSD, developed interacting partners. We had the crystal structure that allowed to model the effects of sum of one mutations and on the protein and monitor its function. But it would have been able to also develop therapies for MSD based on the structure. And of course, there would have been a mouse model for developing and checking preclinical therapies available by the time. And indeed, the group of Andrea Ballabio from Italy, one of the co-founders of the gene, developed a gene therapy. By the time it was published in 2010, they already used an AAV9 vector, integrated sum of one, and then went for a combined intracerebral and systemic gene delivery. And as you can see from this graph, the actual presentation changes a bit over time if you compare it to actual graphs. But anyway, you can see that the sum of one knockout mouse has a really, really short survival. And this is the lifespan curve compared to wild types. And then they went for 3 different treatment options. So they either did systemic gene delivery, only intracerebral gene delivery, and a combined treatment, which was effective to restore or to actually extend the lifespan of these mice. And hopefully you can see from these videos, so this one takes a bit longer to go on, that there was also phenotypic changes in this water maze test. This is the sum of one knockout mouse. This is the sum of one treated mouse with a combined treatment, and you can compare both of them to the wild-type mice, and you can see that there was definitely improvement of the presentation. So I would say at 2010, there was kind of a therapy was on the horizon, but then of course, it takes different milestones to really achieve a therapy. And as Becca already mentioned, and now I mentioned you twice, Emery, in the audience, the driving forces behind this were Alan Finglas, who founded MSD Action Foundation in Ireland, and Emma Olsen with United MSD Foundation, because they were pushing for more therapy approaches in MSD as patient advocates. And this led to the very first International MSD Conference in Dublin 2007, where patients, parents, and scientists met to actually outline a strategy for developing therapeutic approaches for MSD. And from there, we are still moving on. So during the conference, and even later on, we agreed that it would be good to have better mouse models. So the knockout mouse is good, but it dies early. And so the need for a sum of one knock-in mouse or knock-in mice resembling the disease, which is mostly caused because of sum of one missense variants, would be a chance, to mimic the disease a little bit better. We also developed a zebrafish, and we might have an in-depth discussion about this later. There's a sum of one Drosophila knockout model available, and then work that was done here in Philadelphia was the development of iPSCs from MSD patients, and there's a huge collection of MSD patient-derived fibroblasts available. At the same time, therapy development already started for MSD with target identification for drugs that could be used, or at least at all druggable targets for MSD. There was also attempts to do an untargeted drug screening, 2 approaches for gene therapy. So a different group was picking up the AAV9 gene therapy again, and there's also efforts to win an ex vivo gene therapy. Attempts to do hematopoietic stem cell transplantation are ongoing, and there's a project using the FG protein and the crystal structure to do fragment-based drug development for MSD. And during the rest of my talk, I'd like to focus a little bit more on the drug screening, because this approach gave recent results, so we used an immortalized MSD patient fibroblast line and went for aryl sulfatase A activity as a readout in a 96-well format. As you remember from the very first slide, this is one of the sulfatases that is actually reduced in activity, but also one of the most important if it comes to clinical presentation of the patients. The readout was the rescue of the absent aryl sulfatase A activities, and we were able to use a library of 785 FDA-approved drugs that was kindly gifted by LifeArc, so a charity organization from the UK. And the idea was that if we used FDA-approved drugs, we would be able to go for drug repurposing as the fastest approach towards any therapy. This is the usual plate layout, as you can see here, with some controls. And in every well, we had one reaction and one drug. And the readout could be plotted on the right side on a graph like this, where you have the optical density, which correlates to the aryl sulfatase A activity in the spectrophotometric readout. One circle represents one drug and one well, and we have some positive controls and a negative control to determine the range of activity. And indeed, we were successful in finding one hit in this screening assay, and this was tazarotene, which is a third-generation retinoid. I would have gone with something else, honestly, but as this showed up as a hit, we went on and kept on following it. So next steps that we undertook was that we checked if it was increasing aryl sulfatase A activity outside of a 96-well plate, and it actually did in a time-dependent manner where there was a clear increase of aryl sulfatase A activity over time. It also was a clear dose-dependent increase of aryl sulfatase A activity, of course, all in vitro. And as we wouldn't rely on fibroblasts exclusively, we went for the newly designed iPSCs and derived them into NPCs. This was also work that was done here at CHOP, and could see that even the controls were reactive, but of course MSD cells were reacting, and there was a clear increase of aryl sulfatase A activity. So at least we're getting closer to the cells that would definitely need FGE function and sulfatase function. This was not only restricted to aryl sulfatase A, if you want to ask this question, so it worked for all sulfatases that we tested. So it was a multiple sulfatase restoration, which was good. And it was working across different mutations as long as there were missense mutations. And of course, we were not only interested in seeing an increase of sulfatase activities, we wanted to see actually if this was doing something to the cellular pathology. And there's a nice readout that you can do if you stain lysosomes with LAMP1, which is a lysosomal membrane protein, also in late endosomes. And you can see in an MSD fibroblast, the cell is full of lysosomes and late endosomes. And this is the typical phenotype of MSD cells. If you compare it to a control fibroblast here, this is all non-treated conditions. And if you treated these cells with tazarotene, nothing much happened for the control, but you might be able to recognize that this looks more like this or that and not like this. And this is definitely a response to the treatment in vitro. And another really interesting observation was the fact that the treatment was also able to reduce glycosaminoglycan content in the MSD fibroblast. So a hallmark of these diseases are the substrates that are stored in the lysosomes, and especially GAGs. We used a mass spectrometry-based method to look at different GAG subspecies published by Fuller et al. And this was done by Mike Gelb at the University of Washington. And here you can see one subspecies. So this is the peak at the retention time without treatment, and upon treatment, indeed, it's the same peak. And if you calculate this and do a proper readout, you can see that all GAG subspecies could be reduced upon treatment in different MSD fibroblast lines, two with the same mutation, one with a different mutation, all homozygous. This is a little bit milder, but there was treatment response. So overall, we could say that there was definitely an improvement or even a cure of the MSD pathology in vitro, and from there we would like to move on, because next steps on the horizon for the MSD therapy development could well be clinical research projects. So with the tazarotene story, we made it up to become a demonstrator in the so-called Remedy for All consortium, which is an EU-funded consortium and made up of 23 participating universities and stakeholders dedicated to drug repurposing. And as tazotine is available on the market for a different indication that might be well amenable to drug repurposing, and we were selected as one of the demonstrators and could move the project forward. I did not talk much about the SUM-of-1 gene therapy that is also pretty successful in the preclinical models that we have now using AAV9. And if we are lucky, at least we are able to hand in applications for the Bespoke Gene Therapy Consortium call for abstract, because MSD was among the 14 selected rare diseases that they asked for applications, and this is mostly done by Rebecca Ahrens-Nicklas here at CHOP. So to summarize, the therapy development, we went for different MSD disease models meanwhile. Projects for preclinical therapy are in development are in progress, ongoing, some close to be finished. First clinical trials are in the planning phase and hopefully might be launched within the next future. Still, we have funding opportunities through patient organizations, and maybe people are encouraged to join this little group of researchers because there's still more work to do. Thank you very much.
Thank you very much. Far be it for me to keep everyone from lunch, so I both will start and end the morning session. It has been one of the great pleasures of my research career to be able to work with Becca and Lars and Amber and Alan, and this is a ultra rare disease that we have made incredible movement on in the last 5 years, and it is just a delight to be able to be part of it. I think, as Becca outlined, one of our strategies is to understand everything we possibly can about this ultra rare disease. And although, the number of patients we believe to be living with this disorder to be very low, a very large percentage of them are enrolled in our natural history study, which is incredible. And as we are launching our prospective natural history study, a large percentage of them have already participated and have begun to participate. And so while it's rare, we have an incredibly motivated patient population. We do worry about ascertainment bias, and as we learn more and more about this disease, the common theme is we understand one phenotype of each one of these leukodystrophies, and the next generation of children we identify have that exact same phenotype. And as we get smarter and wiser and start to rely on less targeted testing, we're realizing that we have children that are much older with MSD, that are living for decades or have mild autism. And so understanding the spectrum is important, and then going back even further of identifying biomarkers or clinical variables present very early in life that can help us to predict outcomes is going to be essential, both for counseling families as well as designing future clinical trials, because we only know the information we have now. But I suspect that as we get smarter, we're going to be identifying kids earlier and earlier. And that's what the graphs in the blue show, where I plotted the subjects we have enrolled in our natural history study by birth year, right? And then on the y-axis, the time to diagnosis. And so we're getting much better. Families used to wait years, decades, before they got a diagnosis of MSD, even though we have biochemically known about this disease for a long time. But we're now in the range of 1 to 2 years before a child gets diagnosed. It isn't perfect, but it's much better than we were. I was expecting on the red graph for it to change in a way where we were recognizing a bigger and bigger spectrum, and we aren't, which is peculiar. I think there are two things that are factoring in it. We still have ascertainment bias, meaning we're still identifying primarily the youngest onset, more severely affected children. But I think the other thing is, as we're looking into the natural history of these children, we're getting smarter and smarter at identifying the very subtle underdiagnosed features that maybe you only had very subtle developmental delay, and so your neurologic presentation was at 5 or 6 years of age. but your feeding difficulties or your respiratory issues or your recurrent ear infections actually happen very early in childhood. And so I think one of the things we grapple as a rare disease community, and I say this for all leukodystrophies, is understanding what presentation is, right? As we're getting smarter, we're pushing that presentation or that moment of onset to earlier and earlier, and it's becoming a murkier age at which to define. I think the other thing, and this goes to Francesco's talk, and Francesco is helping us with MSD as well, is our outcome measures are largely built on cerebral palsy, right? And those kids don't look like leukodystrophy kids. Maybe we can capture one moment in time of what function is, but we really need outcome measures that are validated in our unique populations, because otherwise, how are we going to longitudinally follow how children do? And in MSD, we find many children have profoundly affected language development, but their motor skills are much stronger, right? But once they start losing their motor skills, they still have measurable disease, but they're not as measurable by our traditional scales. And then, as we have talked about several times now, MSD is a multisystemic disease because it is MLD plus 5 MPS disorders plus ichthyosis disorder and on and on. And so we need scales to measure the dysfunction in each one of these organ systems, but ideally even one scale to reflect MSD as a whole so we can overall measure the burden of disease. This is work from Lars's group where they pulled out every single published case of MSD and started categorizing the words that were used to describe the systemic dysfunction and then bend them into smaller and smaller categories. And so you could really see how the burden of disease, each child began developing more and more organs that were involved the older that they got. And you can start to see these early patterns. With Lars and Becca, we did a similar thing in our natural history study where we looked at the existing medical records and started to identify when children had cardiac problems, hearing problems, neurologic issues. And so we actually published these in the same issue of the same journal in parallel, one being pulled from the retrospective records and the other one being pulled from published cases. Again, giving the story that MSD is systemic and that children have a growing burden of systemic and neurologic disease. There's precedence in rare diseases for developing disease-specific scales that span multiple organ system involvement. On the left in the, appallingly small font size, you can see this is the MPS systemic physical symptoms scale. And so this is measuring, okay, if a child has MPS, how bad is their hearing? How bad is their cardiac disease, their respiratory disease? And so while, yes, you could have a hearing measure, and you do, but if one child has a hearing problem and another person has a cardiac function and you have a treatment that helps the overall burden, you wouldn't be able to measure improvement across a population on just one scale. And we did a similar thing with AGS where we looked at neurologic burden as a whole, so a totality of fine motor skills, cognitive skills, and gross motor skills, and demonstrate that that helps us better understand neurologic function in AGS. And so we sought to do the same thing in MSD, where we created a neurologic subscore, which is based primarily on our AGS symptom score, right? How severe is the overall disease of neurologic dysfunction? And then also systemic subscore, where you can start to count how many organ systems are involved in a child. And the higher the score, the less dysfunction a child has. There's some precedents taking the beautiful MLD-related neurologic function scales, so the GMFC-MLD and the ELFC-MLD, which are shown on the left, and we mapped out in our natural history study how these two scales performed. Again, MSD encompasses MLD, so I think it's a fair use of this scale in our population. And you can see that neurologic function declines over time. And I'm going to point out with my arrow here, it really is this toddler age that I think would be the ideal target for a future clinical trial, right? These are children that are walking, oftentimes with handheld assist, and then they have a very precipitous but predictable homogeneous decline in their neurologic function. Those, I would think that if we could develop a treatment that could help them, we could demonstrate stability of disease. And I'm excited to potentially try tools like the GMFC MLD in this population. Whereas the language function, which you see on the bottom, many of our children gain one single word, but they're not really developing more complex language function. And so any scales or tools in MSD really should reflect the nonverbal communication skills rather than verbal communication, because that's not a strength in the population. When we take our MSD neurologic subscore and we apply it across the population, you can see a similar trend downwards, where over time children are losing function, and we can use it longitudinally. This is in retrospective records, so this isn't even a prospective application. And here's an example of where we took the same retrospective population. We applied the total novel pilot score, the neurosub score, and the systemic sub score. And the spaghetti plot represents us scoring each individual encounter in a child's medical records. And then we triaged it based on overall severity. So we used two different markers of severity. One is genotype severity, which is shown in the middle panels in the row. And then the bottom one is a marker of just overall clinical severity. And it trends. So I think our pilot scale does a moderately good job at trending neurologic function and systemic burden over time. And it is clinically proportional, which I think is important as we're doing scale development. So what do we do now? A lot of things. As Lars and Becca hinted to, we're really at the beginning of a journey. We have launched a prospective natural history study, which is funded by our patient advocacy groups and newly funded by the NIH. We're going to be monitoring systemic complications of disease so we can get not just what's reported in the medical records, but really what's the rate of cardiac dysfunction, liver dysfunction, et cetera. We're going to look at validating outcome measures and seeing how they perform in our unique patient population. We're going to iteratively refine our MSD scale, apply it to this population, both retrospectively and prospectively, and we're going to collect biomarkers by MRI, CSF, and blood. Oh, and I left off urine. And so in conclusion, MSD is rare, ultra, ultra rare, but because of amazing patient advocacy groups, we have really collected an extraordinary amount of understanding about this disease, and I think are well poised to go into the next natural evolution, which is treatment exploration. And I'm really excited, and hopefully maybe not the next GLIA meeting, but in the future GLIA meeting, we'll be able to present some preliminary data on clinical trials. We can use these retrospective scales to map neurologic function, but I think we can do better and create disease-specific scales that help us. And I'd like to thank the Family Foundations and Amber, who's here, for funding our natural history study. So our acknowledgements are extensive. We work very closely with Lars's group, who is also a site of our natural history study. We work with Mike Gelb's group in biomarker development. Maurizia Pascali, who is head of ARUP, also is helping us in biomarker development, and an extensive group of collaborators around the world. And first and foremost to the MSD Family Foundations and patient advocacy. Again, although it's ultra, ultra rare, our participation in our studies has been extraordinarily strong, and that's because of our amazing family foundations. So thank you very much.
If I can invite the work group down, and we can take some questions because we have a little bit more time before lunch.
Thank you, ma'am. Laura, I wanted to ask, as you know, I'm particularly sensitive about delay to diagnosis. Are there any lessons learned besides apparently having an Amber in your camp? Are there any lessons learned in terms of how do you decrease that time to diagnosis that probably would be relevant for all of the other disorders that you know, all the rare disorders in general?
Thank you. I think that's the time to diagnosis, I think, is going to really limit therapy development, because we all know that window of opportunity to make the biggest differences at the very beginning. And in the end, I think it comes down to newborn screening. I think that's the way we remove our ascertainment bias, you know, who we're actually thinking to send the testing on goes away if it's part of newborn screening. And in MSD, we're really lucky because we can leverage to ongoing newborn screening programs because it does affect other disorder biomarkers as well. And so while probably MSD-specific newborn screening wouldn't happen because of its ultra rarity, the fact it does affect other biomarkers. And we've spoken with several of the newborn screening programs, and once they're clinically acceptable, they're willing to read out on MSD. So I think that that's going to be a really exciting area for development. I think the more I work in rare diseases, the more I realize I can't bias myself on what I think a disease looks like. And that's a lesson I think everyone here in this room knows. I don't have any lessons other than keep an open mind and test for it.
Hi. Okay. My question is related to what you just mentioned about the newborn screening, because there's so many rare diseases. So how many newborn screenings can be done to a newborn? And some of them are not genetically related. So how do you decide?
Yeah.
State has to get their own approval.
Right, exactly. So within the US, even though there is a recommended uniform screening panel, every state has to choose to implement it. The beauty of biochemical disorders, and I'm going to make a plug as a metabolic physician, is that adding on more mass spec-based assays or mass spectrometry-based assays, it is actually not like you have to draw more blood from the baby and it's just a heel prick test. So adding more tests to the sample is actually, while technically challenging to develop those assays, and Mike Gelb, who's helped us, has done an amazing job doing a lot of these assays, it's not more burden on the patient or the babies that are born in the, but it is really important in terms of those early identifications that Laura was speaking about. And so adding specifically for MSD, I think that the newborn screening for MLD will be the best bet in terms of capturing these kids. So it's just a matter of, if MLD screening is widespread, making sure that the state labs report that out. Because today in newborn screening in the United States, right, you can pick up a lot of diseases that don't have therapies because you see the data from the mass spec-based assays. And so the question is, do you report that or not? And every state's a little different. I'm not going to get into that. But once there is a therapy available for MSD, it's going to be really important that this be added and read out.
Hi, Lars, nice talk. I want to ask you about the compound. What type of high throughput screening you do?
So we used an ordinary aryl sulfatase A activity assay. This is a really old method. It's a spectrophotometric readout where an artificial substrate is used, and this is a sulfated substrate, and then the sulfate is cleaved off by aryl sulfatase A. And it's specific if you add some ingredients there for aryl sulfatase A, and then you have a color change from yellow to orange, and this is how it worked actually.
Okay. So the assay, did you check other sulfatases as a counter screening assay?
Not directly in the counter screen itself. So what we did for counter screening was, of course, we checked it without cells, and to see if there was any kind of reaction directly with the compound. So we analyzed the hits further on for all sulfatase activities, and we were lucky. We didn't end up with 200. This was a couple of hits, and so we came down to the one that I was just showing.
What the target is?
We're working on it, honestly. This is kind of the beauty still on an untargeted screen, because you put something on cells and you get a readout, and the rest in the middle, is up for proper research. What we know so far is that you need any kind of residual function of the FG protein. It's not working when you have complete knockouts or missense mutations that are not compatible with any gene, with any protein expression. Fortunately, this only accounts for, I would say, less than 5 or even less than 2% of the MSD patients. They are also presenting with the most severe form of the disease, probably not amenable to even in day one treatment because of intrauterine problems. So we are still doing the right thing if we develop something for the rest.
Okay, we're going to take one more question.
Yeah, this is hopefully quick, but as the follow-up on the tazarotene story, do you think it's working through the retinoid pathway or probably independently? Because presumably you would have picked up other retinoids in the FDA screen if
No. Yeah, it's yeah, it's delayed me, yeah. There you are. What I'm also able to disclose as an answer is that if we block the retinoid acid receptor, we didn't see a response. So it's working through the actual retinoid acid receptor mechanism.
All right. Thank you very much to both of our late morning speaking groups.